This will be more of a general talk, and I think it may also be a little bit of a very elementary nature to microbiology people, particularly those who are working on plant microbe or any microbe interactions. Maybe very elementary level talk. I'm not trying to make very technical. Some of my own research data, some internet collections, some philosophy, as I always do. There are also hypotheses. It will be maybe only 30 minutes talk. Okay. This you already know. The hidden part of plant is mostly, it's not root actually. That's the first thing. When you say root, it is not root. About more than 90% land plants are mycorrhizal. What they have, this is the main roots. This is also a roots. This is also roots, but they have a huge network of hyphae. And these hyphae are inside also. Inside, if you cut a section and see, you'll find different structures. Some hyphae, some arbuscular structure, flower-like structure, some vesicles. In fact, reproductive particle through which they multiply, they produce more hyphae. And this plant has been there for maybe almost 2 billion years. These fungi have been there and then have tremendous role in plant growth and development. One major role is that those nutrients in soil which move slowly, phosphorus or some micronutrients, just a conceptual thing you might see here, that when there is a root, it absorbs nutrient, for example, phosphorus. After some time, this part will be depleted of the nutrient. And the concentration of phosphorus at any point of time is very less. And these ions will move by diffusion process, which is a slow process. Therefore, if you do not have mycorrhizal help, that what plant, soil volume plant can explore will be limited. But because of presence of these hyphae, so phosphorus from these points also will be absorbed and will be put into inside the root. So this is how mycorrhizae provide benefit. The other, and that this is an obligatory phenomena, like fungi cannot grow in the laboratory. And what benefit fungi get from, this fungus get from the plant, different kind of food material, root exudates, or inside different kind of metabolites it use. Another, and then as I said, phosphorus nutrition is a major role and also helps in water absorption compared to a plant where there is no mycorrhizal fungi. Presence of mycorrhiza will definitely help water absorption also. Even some people say it also provides resistance to diseases or tolerance to diseases. And this is a very, like, this was my PhD work, one part of my work, small work, that it enhances phosphorus nutrition. This is very obvious, where these are not mycorrhizal plant, these are different species of mycorrhiza. The plant AMF enhanced P nutrition hasten flowering. The flowering comes early. And one major role of phosphorus is to produce, bring flowering early and produce more grain. This was like a copybook experiment. And other important thing in your forest trees, it is extremely useful organisms. You inoculate them with different species, you get much better growth than the uninoculated plant. So another group of organisms that remain hidden, everybody knows, physics, chemistry, biology, everybody knows. And the wonder of this is, it's a major contributor to the protein need. I'm saying protein need, actually nitrogen need. Earth's whole, the surface and the aquatic system, ocean, it fixes this many million tons of nitrogen. And symbiotic nitrogen, like rhizobium, brings about 120 million tons of nitrogen every year to the earth. You can imagine if they were not there, what would have happened. And if you look at this data, industrial fixation, this is I think 2005 or 6 that are older, I have not updated, I collected, just picked them up from some previous presentations. Only 65 million tons of urea is brought to the industry, nitrogen is brought to urea making industry, right? So microbial contribution is huge. And if we do not take care of our soil, if you grow crop 3 or 4 crops in a year, like in some area of Assam, even those are inhabited by Bangladeshi people, they grow 3-4 crops, and in this, and use a lot of fertilizer, pesticide, you do not see enough of this rhizobium, whereas the traditional way, when the crop grown in Brahmaputra Valley, you get large number of nodules, so naturally they are enriching the soil with nitrogen. Let me go to another group of organisms, which now the current interest is. First I start with the chemistry people, like quite often you make a present, present this, and I sometimes ask question also. The diffuse double layer. Whenever you have a colloids, negatively charged colloids, keep in a solution, electrolyte solution, then positively charged ions are attracted, and then gradually you find a distribution pattern, and finally a region where it is the positive and negative charged ions are same. Right? This is what the electrical double layer concept also you find bovine serum albumin he has shown in the past. Now see what is happening in the plant. In the root region, like every human being excrete a lot of material, like each one of us, if you look at this hand surface, there will be large amount of metabolites coming all the time. Now similar is the case with the plant, large amount of metabolites keep coming all the time, and a soil where you put a plant, moment that plant comes, it start excreting those, and the soil, the microorganism that is residing in this place, they will orient towards the root, but that there is a selective pressure even. All microorganisms present in that soil will not come near it. You'll see that in a short while. Now they also form a gradient, obvious, because if you look at these roots, they secrete a lot of carbohydrate, fats, proteins, and put an iodine solution, you see the distribution of, you get a idea how these plant root metabolites are spreading, particularly carbohydrate. This is up to a certain distance it goes, and that's why the bacteria also orient like that. If you take a photograph, these are all full of bacteria around. If you take a aniline blue stain on the root surface, you see bacteria, and scanning electron microscope, you can like see how bacteria are present there. Now let us see one published work. In 2005 it was published. You take one soil, same soil, microbial composition, everything is same, and grow 5 different crops in replicate samples, and you take the rhizosphere sample, rhizosphere soil. I will come to that rhizosphere soil collection as a problem as such for all people who are working on rhizosphere microbiology. Now what you see here is a disease denaturing gradient gel electrophoresis. I need not explain, you know this institute does quite a bit of work on that. Now what you see here, in the same soil, you grow different crop, they bring different kind of, in the same soil, all the bacteria are common, but it selectively bring only a few particular species. This is what is reflected. Just I will give one example. Veronica, this species, you have this band. Each band you can assume as those who are not very used to disease and microbial profiling, DNA. For them, assume these are individual bands are one bacterial species. Now this species is not here, but it is in the soil, but it is not here means this has not, it not got attracted to us, this plant doesn't need it. So there may be some amount of specificity controlled by the plant. Now whenever we do research on rhizosphere microbiology, it is easy to say like that metabolites spread and microorganism also show a distribution. But in reality, when you want to collect samples and see the distribution, it is tough. And there is no any mechanical or automatic device yet. I always had that problem. So what we do, taking utmost care, we take out a root. And those which are not attaching to the root means we treat them as bulk soil, like you saw the bulk in diffuse double layer, because in here, this is a natural soil with all the original microorganism present. There is no influence of the root. And the other one, like you have the soil is adhering there. These are the rhizosphere soil. Even if you give a very strong jerk, then you find some loosely adhered soil come out, and some soil are difficult to remove. So we say them as very strongly adhered soil. Remove them, surface sterilize the roots, and then what you find, you'll find bacteria inside also. So that way, bacteria, 4 types of bacteria can be delineated, isolated. Now, let me, before I go further, give you a beautiful summary of what has happened last 10,000 years or 8,000 before Christ. Now this agriculture, when it started traditionally, what happened? You get a definite level of yield, but every year, if you take into account the loss is always there whenever I do. So there is some loss, and that loss actually, the grain that is formed, the plant feed the insects, whom we say as pest. But insect, and it was very balanced way. Every year you some amount of crop loss you get, right? And what happens to these two graphs show the contribution of below ground microorganisms present. One I showed mycorrhizal response or agricultural natural nitrogen fixation. So in crop production, these microorganisms historically has been contributing tremendously. Come 1950, human population started increasing, our nitrogen consumption went up, nitrogen, phosphorus, and even pesticide use increased. And as a result, we brought more insect attack, more loss due to attack insect and pest, right? And then the contribution of the natural agents has declined. So overall productivity of soil has gone down. Now, like our target now, by 2050, their contribution we must increase. This is where is importance of below ground diversity of microorganism in production. And like applying fertilizer, you are not going to achieve increase in production is because if you see this agro-vegetal production and fertilizer is this is do not take this an absolute value. This is an index. I now forgot to read this paper about 4 or 5 years ago. Index means what does this means? Agro-vegetal production and fertilizer use. If you see that for every this many million ton, you get this many production, and now for this many million ton of fertilizer, you get this many. If you divide these two, per unit of fertilizer used, like loss now is much more. It gets lost from the system. So alone by increasing inorganic fertilizer, you are not going to be able to, unless you take the soil system as a natural resource, and all other components that contribute to growth is also taking part in the production process. This was one experiment that when I was there in Assam Agricultural University, I could do only one experiment very thoroughly, about 5 years' time. It's again copybook experiment. Now, Assam, like there have been effort to grow 3 crops in a year, and this is in old days people used to do that. You grow 2 rice crop, and in December month you sow pea seeds. Now, if you grow 3 years continuously, say like rice, 3 years you will have 6 rice crop and 3 pea crop. After end of 3 years, where you don't apply fertilizer or organic manure, you cannot harvest anything. Means the soil is totally gone. But if you keep using fertilizer, you are getting yield. And another very interesting thing here, fertilizer produce more of straw than the green. And this particular practice, I'm not going to show all details that you have to only believe me, this is published already. It produces less straw or biomass, but more green. So it's a balanced integrated nutrient management means we have put 2 group of bacteria, bacterial culture, along with organic manure. Another experiment when I was in IBSD, like what we wanted to do, that was already discovered by somebody, so we resorted to study on mandarin orange. You see, bacterial isolates have individually, they differ in enhancing plant growth. The control compared to control, some of them bring very good seedling growth enhancement. This variability is always there on this below ground community. Now I think this graph alone doesn't make a lot of sense, but what I'm trying to tell in our experiment, what we tried to do by your NGS sequencing, like this is not NGS sequencing, those DGG band we cut, amplified further and sent for identification. What I'm trying to show here is that some bacteria, they will be present in the bulk soil, they will be present in the strongly adhered rhizosphere soil, they will be present inside the root. And some bacteria, and this also what we did, this particular 3, 6, 12 months sampling, means we did a destructive sampling in 3 months, 6 months, and 12 months. This 3, 6, 12 means in all the sampling time, these 2 bacteria were always these two bacteria were always there in soil, and means they were moving from soil, perhaps they move. But if you look another bacteria, this particular bacteria, they remain only inside the root. Maybe they are in small quantity in the soil, from there only, or maybe it was with the seed. Okay, so what you see, like there is a distribution. Some bacteria all throughout, and some, why this happens, there are some of the questions we need to understand. So some of the research we're doing now. Another, again, to glorify microbial research. In 2000, this also some of you have seen, Circa Award 2010, it was a Google competition in which 3 Ireland girl bet, even they were outcompeted some beautiful models of your aerospace and all that kind of thing, sophisticated thing. Why? What they did is fantastic. After, like, just read this. After 3 years of treating more than 13,000 seeds, concluded diazotrophic bacteria treated seeds germinated at least 50% faster than the untreated seeds and increased yield of barley and oats. They are only high school student. And if you go to internet, I had another slide I didn't pick up. The website was there. You can go through this. It's very inspiring. And how much trouble they took in their home, in their school, everywhere they kept sample to test. Okay. Now our current focus. Here, and you have been hearing my students a little bit deal on that. What we want to do, current focus is, because rhizosphere is a little bit of complex. We want to get into a very pure system. Seeds inside is easy to work. So we are taking 3 crops: rice, green gum, mustard, and Sadav is working on tomato and potato, tomato and tobacco. Like someone yesterday, I could not contact him. Now, what we want to do here, the spatial distribution of the endophytic bacteria in seeds, roots and shoots of seedling, like when it germinates, the root and shoot come out. Then successional pattern, specific group proliferating at specific time and helping plants, means all these bacteria present there, will they like proliferate depending on what stage this plant is? Or all of them proliferate at just start of germinations, some of the questions, and how they are spatially distributed. And already one group from Germany or Netherlands, Netherlands, they have already shown that first work, that there is a core group and there is a transient group. That also I'll come very briefly. Now this is, I'll give the example only with gill meal work, but we have data for other crop too. You have a surface sterilized, you have some surface sterilized rice seeds. On the surface you don't see, these are some image, you don't see bacteria. But if you cut a traverse section, you see bacteria there, right? And if you grow these seeds up to 5th day, the seedling, you find bacteria inside this and inside the root. And those 5-day seeds, your seed doesn't dissolve completely in 5 days' time. Now, on 5th day, if you look at the surface, on the 0 day, on the surface was clean. On the 5th day, you find in the surface there are bacteria. So, the bacteria which is present in the rice, and these are repeated 7 times, as Jill will 10 times, as Jill will say, in 10 different seeds she found it. So, from the inside, the bacteria again come to the surface. Now, next stage was, this is very preliminary result. I just insisted Jill Mill to give me this data, whatever way she can. So, what we did, this experiment is a very extensive experiment. Like Jill Mill will present later, means during her thesis progress presentation. Next generation sequencing based bacterial community. This technique you already know. When you get DNA, you are getting DNA of the seed origin and also bacterial origin. Amplified 16S RNA gene primer, and what you are getting is all bacterial origin. Then sends for sequencing. And these seeds, just 0 day surface sterilized seeds, 3 replicates of 1 gram seeds. What you see here is, again, each color is representative of a particular phylum. Okay. Now here this is Streptophyta, I'll come to that, and this is Protobacteria. Now this is the fifth day seeds. These seeds are different lot, not the same lot, because this is gone. These are destructive sampling kind of thing. And here what you find, entire seedling DNA was taken, and what you find that this particular group it's diminishing, getting reduced, whereas these Protobacteria, they are increasing, and all 3 replicates you get the same result. Now this set you grew for 15th day under aseptic condition, and from root, these are 3 replicate root samples. What you get is Protobacteria in large number, and the shoot, the same thing. This is very preliminary to say, but I mentioned that whether something is happening, that at each growth stage, the bacteria that are existing inside the seed are probably like increasing in number, not at one time that they all, and succession, but you all know, is a rule of nature rather than exception. The succession is a big phenomenon in microbial life in nature. So as we go through, maybe in future we'll hear a lot of new things. We hope we will hear. Now let us look into another aspect. You have seeds here. These seeds already have this is again hypothetical kind of thing based on their work, Hardroim work. You have endophytic bacteria already present inside. You have 2 soils where microbial community is different. Now you grow it, the seeds are again developed. In the next generation seeds, they have this. In addition to that, you find 2 other species or 2 other type of bacteria. So this is what Hardroim says, that there is a transient group, means from this soil, these 2 is picked up, and the same seed, means the same rice seed, not same seed, like rice seeds again grow here. You have the original rice seeds endophytic bacteria, but from this soil it has picked up too. So depending where you grow a crop variety and the composition of bacteria in that, your community may change. This is what is already a published report, the first published report in fact. Now let us ask a question. As I said, there will be some philosophical thought. When did the endophytic bacteria enter into plant and its parts, including seeds? First, I will go not to these wholesome bacterial cells, but to a endosymbiosis hypothesis. Many of molecular biology students and microbiology students, you might know it already. So about 4.5 billion or 5 billion years ago, our Earth evolved from present time, and these pyrocetes were the first organisms, the protobacteria. And then what is said that these bacteria entered into primitive eukaryotic cells, and they become chloroplast and they become mitochondria. And this is what is when microorganisms become part of plant, but our work is not related with that, but I'm trying to give a historical perspective, what we know now. Our plants evolved somewhere here, rice or maize or whatever plant you say. Now these wholesome bacteria, because these prokaryotes, when it become part of eukaryotic cells, I'll go to next slide. And endosymbiosis hypothesis, like everybody, all scientists agree to it, about 2 endosymbiotic events happened approximately 2.7 billion, 2.7 means 270 crores years ago. What happened? You had protobacteria, it entered, this is as a mitochondria. In the second event, like cyanobacteria entered, becoming chloroplast. And today, in higher plant cells, you get chloroplast, which lock energy from sun as ATP and food, and unlock this particular structure, mitochondria, it unlocks energy for growth and development of lives. This basic 2 process that where we survive happened 2.7 billion years ago. And like there are, in this hypothesis, there are a lot of logic and argument. Mitochondria and chloroplast and bacterial DNA are circular. Then chloroplast looks like cyanobacteria, double membrane cover, evidence of engulfing, means through the cell membrane of eukaryotic cells, these prokaryotes entered, they have their own membrane, and also this membrane of the plant also they took, so it is a double membrane. That's another evidence. Then mitochondria contains about 37 genes, out of which 15 similar to bacterial genes. Chloroplast contain, definitely chloroplast has much bigger role. Making is always easy, is difficult, but breaking is very easy. It unlock, it breaks. So chloroplast breaks, so it has, that's a philosophy. And this is a huge system, the solar energy harvesting. So it has large number of genes involvement. Now if you go through recent literature, there has been huge amount of work saying how chloroplast of that entered from prokaryote has transferred some gene to the nuclear DNA of the plants. A lot of interesting readings are there. So biology students who are interested can go through those. And then another interesting thing, bacterial genome sequencing was like during 1997, 2004, several bacterial genomes were available. And rickettsia, you all know, cause high fever. It's a very dreaded disease at one point of time. You all know that. So this is closest relative of mitochondria. This mitochondria is the closest relative. Can divide and grow naturally within eukaryotic cells in ticks, mites, which spread the disease, and also can briefly escape, not for a long time, briefly it can escape and again enter into the host. But this mitochondria and chloroplast has lost this ability to come out and multiply. They are not autonomous. They need the support of the plant, that you all know already. Now then, our question now, when did the wholesome bacterial cells become endophytic and for what function? Our question. This our bacteria, when did they come? Like not much, like not many people have asked this question. Maybe people ask this question, but we have raised this question that when did come and can plant function without them? If we remove them, can plant function? Will the plant be able to function? Let us again take that, this is already I have explained, but I was going to, let me show what happens. This is coming. So this is how, like whatever is existing, it will go to the seeds, from seed to seed. So from soil also some bacteria can go, right, and become a transient group. This is what one scientist says. So we want to ask, how about, because these spikes, these pistils, and those stigma, pollen, they are all exposed. So can bacteria from the air go and enter into the seed? We have been trying to devise a system, completely closed system, where even it big enough. From a seed, we grow a crop, rice crop, grow it, and then it again produces the seeds. Okay, one will be totally sterile environment, and one growth media sterile but exposed to air, and then one growth media of unsterile soil and closed system. And this is soil. So from soil, some transient group will grow. From soil, some air transient group will go. If we compare, like these are all hypothetical things, but hopefully students have enough material to think and carry out research. Another thought that came to us, that we grow these seeds, first generation, and this will be grown with Hoagland solution in sterile media, and we produce seeds, these seeds, and these seeds, from these seeds, all the bacteria here and here should be very uniform because there is no chance of getting any transient group. And we continue doing that several generations, 3-4 generations. Whether all the bacteria will be retained, or if they are really essential, obligately dependent, they will be retained. If not, some of them may get they escape. Then we'll know that whether this association, now chloroplast and mitochondria cannot escape, but can they escape? Because we do not have direct answer when they entered and why they entered, but this can be, there's a lot of, we have been last 3-4 months, we have been with that Borosil people, we have been trying to develop this system. It's not coming out nicely. Now I think I have finished my talk. Only again I bring you back to what Professor Datta said, is what I have used, abstract, elegant expression that connect beauty of creation. Whether it is a leaf, or whether it is a beautiful or ugly girl. He said that same golden ratio. Now, this graph also, what is expressed, this graph also expresses human efforts and interaction with nature in food production process, bring to focus microbial role. With these words, thank you very much.